The National Vaccine Development Roadmap, or PPVN, and Malaysia Genome and Vaccine Institute, MGVI, were launched by the Malaysian Prime Minister, Yang Amat Berhormat, Dato Sri Ismail Sabri Yaakob, on the 1st November, 2021. Through the implementation of PPVN and establishment of MGVI, Malaysia could produce its own vaccines that are of quality, effective, and safe in accordance with the conditions set by the National Pharmaceutical Regulatory Agency, NPRA. A vaccine is a biological substance designed to protect humans from infections caused by bacteria and viruses, through stimulating the production of specific antibodies, and thus providing immunity or protection against the disease. Vaccination has been undeniably very helpful in promoting a healthy global population. It has greatly saved lives, reduced disease burden, reduced healthcare costs, and in general raised the overall quality of life throughout history. Conventional vaccines, for example, attenuated or inactivated vaccines, are made using actual viruses or viral components, and can take several years to successfully create, due to the process of collecting the viruses and adapting them in the lab. That changed with the COVID-19 pandemic. The exponential rate of disease spread seen in early 2020, and related case projections demanded the rapid development, evaluation, manufacturing, and deployment of successful vaccines, leveraging on decades of progress on new vaccine platforms, viral immunology, structural biology, protein engineering research, and bioinformatics. Bioinformatics is defined as the application of tools of computation and analysis to the capture and interpretation of biological data. It is an interdisciplinary field, which harnesses computer science, mathematics, physics, and biology. With the development of bioinformatics, it has been possible to construct extensive databases of various biological quantities such as DNA, RNA, and protein sequences, analyze the huge quantities of accumulated data for similarities and dissimilarities to understand newly sequenced sequences and their interrelationships, and gain new insights. Immunoinformatics, in particular, is a branch of bioinformatics utilizing mathematical and computational approaches to process and develop immunological data, which includes the complete information on an organism's genes and proteins that are involved in an immune response, and make predictions on immunity and disease pathogenesis. The reverse vaccinology model, in contrast to conventional vaccine development, starts with the genomic information of an organism. Reverse vaccinology uses bioinformatics techniques to screen entire genomes of pathogens, to determine genes that could lead to good epitopes, which are peptides in an antigen, to which the antibodies actually bind to and activate specific immune responses. The process starts with genome sequence analysis, in which computational programs are used to identify all open reading frames, ORFs, that encode for peptides or proteins in the sequences. These predicted peptides are then screened in a variety of aspects, including antigenicity, allergenicity, peptide toxicity, surface accessibility, hydrophilicity, and peptide conservancy. This allows selection of good epitopes that are consistent across pathogen variants, more likely to have antibodies bind to them, produce minimal adverse vaccine side effects, and more likely to be effective over a long period of time. Some of these assessments involve another subdivision of immunoinformatics called structural vaccinology, which uses molecular docking, dynamic simulations, and homology modeling to deduce conformational features that could affect immunogenicity. Structural vaccinology is also used to introduce designated features that stabilize the epitope. For example, 2 alterations to prolines in the SARS-CoV-2 spike protein helped stabilize the spike protein in a laboratory environment. thus facilitated the rapid and successful design and production of a high-quality immunogenic spike protein that fueled the rapid development of diagnostics, therapeutics, and vaccines at the start of the COVID-19 pandemic. The type of predicted immune response is also selected for in the screening process, whether it's humoral or cellular. Here, the large set of publicly available epitope databases and information present aid in selection of desired peptides as vaccine candidates, VC. These vaccine candidates are then synthetically produced and tested in animal models and clinical trials. In all, the use of bioinformatics in vaccine development is highly beneficial as it is cost-effective, reduces the time spent in the traditional drug design approach, and more importantly, allows selection of vaccine candidates that are structurally stable, safe, and effective.